Our next speaker is Ko Sugawara. He is a postdoctoral research fellow at l'Institut Génomique Fonctionnel de Lyon in France. And he will be telling us about elephants tracking cell lineage in 3D plus time by incremental deep learning using sparse ellipsoid annotation. So thank you so much. Please take it away.
Thank you, Guillaume. Hello, everyone. My name is Ko Sugawara. I'm a postdoc working at IGFL Lyon. And I really appreciate this opportunity to share our work today. In this talk, I will introduce elephant tracking cell lineage in 3D plus time by incremental deep learning using a sparse ellipsoid annotation. So cell tracking is a widely used technique in various fields of biology. For example, in developmental biology, tracking cell lineage using microscope is a powerful approach to investigate the developmental process at a single cell resolution. Please play the video. So here, I'm showing the C. elegans developing embryo dataset from the cell tracking challenge. As you can see, in the beginning, it is easy to follow the individual cells, but rapidly it becomes difficult to track. So the tracking results can be used to generate this kind of lineage trees. And the important point is that the tracking results need to be error-free to do this analysis, because only minor mistakes can change the structure of these lineage trees dramatically. So it is important to have the tracking system that allows the users to inspect and proofread the results in an efficient way. Please play the video. So this is a tracking result generated by elephant. So by using elephant, we can achieve this quality of tracking results within a day. But in the manual workflow, it would take several weeks to finish it. I will talk about how it works in this talk. So there are several approaches in cell tracking. The first approach is manual workflow, which is accurate but time-consuming. The second option is rule-based algorithm, which is quick to try but limited in accuracy. The third approach is deep learning. It's becoming popular, and it has the potential to achieve an accuracy that can be comparable to the manual workflow. However, its accessibility is still limited. I will explain about that in the next slide. So deep learning has emerged as a powerful approach in bioimage analysis, as you know. So there are many applications like classification, segmentation, augmenting microscopy, or image restoration. Generally, deep learning performs very well, but it requires a lot of annotated data for training. And users also need to work with a script on powerful computing resources. These are the main obstacles in training in deep learning. So the objective of this project is to establish a platform for cell tracking that integrates deep learning with enhanced accessibility. This diagram shows a common deep learning workflow. First, the user will annotate on the hundreds of images. In general, this step can be a bottleneck of the whole entire processes. After finishing annotation, those annotated data are fed into the training module, then where the deep learning models are trained. After finishing training, these trained models are used to predict on the new data. Then, in many cases, those predictions are not perfect, so users need to proofread the results to get the complete cell lineage data. And typically, those steps are divided into different software components, so users need to go back and forth between them. That is not so useful in the practical situation. So on Elephant, those steps are integrated in a single workflow. So the users can start the cycles of annotation, training, prediction, and proofreading with just a few number of annotations. And So yeah, this is just iterating these cycles can generate the cell lineage data efficiently with incrementally trained models. So I will briefly speak about sparse annotation, but Anna already had a very amazing talk, so I will not speak about that much. So basically, in general, rich annotation, dense annotation is performed, but on sparse annotation, only in a sparse subset, a small subset of cells are annotated like this. I will also mention about the use of ellipsoid. Point annotation is simple, but it's limited in the amount of information. On the other hand, polygon annotation can include a lot of information. However, its annotation can be complex, and the number of parameters to handle can be increased depending on the number of vertices. Ellipsoid annotation has an intermediate characteristics, so it has a relatively good number of information, and annotation is not so hard. And the number of parameters is just 9. So that is why I decided to use ellipsoid as this representative of cells in this tracking software. So tracking on elephant is done in 2 steps. The first step is detection. Then the second step is linking. So in the linking phase, the detected cells are associated in time dimension. So here, for example, this green dividing cells is associated with 2 daughter cells in the next time frame. So this a step is called linking. So this can compose the lineage trees as shown here. So in the detection step, user first start with putting a small amount of sparse annotations. Then these sparse annotations are converted to the training labels. So here, the green is the cell periphery region, and the blue areas are cell center regions. So background are colored in magenta, and this can be either explicitly specified, or the intensity thresholding is also working for the fluorescence imaging. Then, based on this training, so the black areas are unlabeled, and these areas are ignored in training. So basically, this is a 3-class UNet model, and based on this training data, the model is trained and prediction is performed like this. Then we extract the blue area to reconstruct the cell shape, as shown here. Then the user proofreads the results and iterate this cycle. I will just show that. For example, here we put 1 positive annotation and 1 negative annotation. Then prediction will give this kind of data. Then proofread it, annotation, then train again, predict again. So basically, every time the library is updated, the model is also updated. Yeah, that's it. Then we tested this method in various types of data, and we found that it actually works on these kinds of data sets. So not only the sphere-shaped, but also we can handle the elongated shapes with ellipsoid. So this is showing the starting from the pre-trained model, putting a sparse annotation and train again, and gives better results. That is a summary of this picture. So in this demo movie, I will show a Drosophila embryonic data from the cell tracking challenge. And yes, so this is actually shows how it works on the very sparse annotations. So this is randomly initialized model, so it gives a very poor results first, as shown here. Then I put just 1 annotation. Then start training. Then immediately after training, it starts giving a very good result as shown here. So it's just 1 annotation. And I'm predicting locally now, but this prediction can be done for the whole volume. So if the data is very big, we can just predict locally. And if there are some missing nuclei, we can put additional annotation. And if there are some false positive detections, we can also put the negative labels as shown here. Then in just 5 minutes, with just 2 positive annotations and 2 negative annotations, we can achieve this quality of deep learning model. So actually, training deep learning model does not require so much annotation. So of course, it depends on the characteristics of the data, I think. So because of the time, I cannot explain much about linking, but the concept is the same as the detection step. So we use the sparse annotation, and instead of predicting the probability map for the 3 class, here we predict the movements of the cells between the time points. So the idea is that if we know that how the cell moves between the time points, we can get a better linking performance. That is the idea. But it also uses unit architecture. So finally, I'd like to briefly introduce about how elephant is used in our research. So this cute crustacean animal called Parial hawaiiansis is our daily using animal, and this animal can regenerate its limb after amputation. So please play the video on the bottom right. Yes. So this is a movie of the regeneration process. So this is established by our colleagues, but so the image quality is still limited. Signal to noise ratio is not so good, and there are a lot of proliferation events as shown here. So I would say that this is a challenging data set for image analysis. But this, please play the video on the right. So actually, by using elephant, we could achieve getting this kind of tracking results. And yeah, we could collect a relatively good amount of complete lineage trees. So we are now starting analyzing these data. So how to get started with elephant? So elephant implements client-server architecture. And basically, client is based on Mastodon on Fiji. So you can just click the update site to install it. Then the server side, actually it does deep learning, can be set up using Google Colab. Or if you have a powerful workstation, GPU workstation, or HPC cluster, you also have those options. So everything, those steps can be found in the user manual. So just please visit here to get it. So finally, I'd like to thank all the people who helped this project, especially John Eve and Tobias for developing Mastodon, such amazing software. And I also want to thank those nice community, open source community, Nubias, and EML machine learning course was very helpful for me. Thank you, Anna. And yeah, that's all. Thank you so much.
Thank you for such a wonderful tool and talk, especially. So cell tracking is always very close to my heart. Any questions from the audience? At least, so there's one here at the front, at least.
Yes, thank you very much. For three-dimensional data, wouldn't it make sense to use a spatial model instead, like a rotational ellipsoid?
Sorry, the question is 3D. Could you repeat again?
Yes, for three-dimensional data, wouldn't it make sense or improve the model if you would use like a rotational ellipsoid, a three-dimensional instead?
So rotate the 3D volumes to before analyzing the data.
Exactly.
So I think it depends on the modality. I mean, so if the isotope, if the x, y, and z dimensions are very different, maybe we need to care about that. But I'm not sure if the rotation can help that. But yeah, we can try that. Thank you.
Okay. Thank you. So we have time for at least one more question. Okay, so then maybe I can ask one. One thing that I'm really excited to try some of those deep learning methods for cell tracking, where the deep learning is used also for the actual linking step. But one thing that I'm always a bit curious about, and it would be great to hear your thought about that, is because now you're literally predicting cellular behavior, and you're teaching the model on predicting this cellular behavior. But now, if you treat your cells with a drug, that changes that cellular behavior. So how well will the model actually work? Because then now your cells will start to behave in a completely different way. So do you think that you can use the same model to predict that, or then the linking step will not work and you need a new model?
So yeah, it's a very good question. So the model is trained on specific time points, and the specific movements we selected. So if we find that the model is not working well, we can put additional annotation on that regions. So that is a good point to do it interactively. If we can find errors, we can put the annotations there. So then update the model and get the feedback. And that is a very interactive process will help to work on that problem, I think.
Okay, great. That sounds wonderful. All right, so I think for the interest of time, I think we need to now move on. So thank you so much again for the great talk. So the